fermentation, as well as cannabis oil-infused cookie dough in mice. So Haley, if you want to share your screen. Perfect.
Hi, can everyone hear me? Great.
Yes.
First and foremost, thank you to all of the coordinators at IBANGS for putting on this virtual symposium. I'm very happy to be here talking to you today on some of the recent work that I've been doing on this project I lovingly refer to as the On Baking and Getting Baked project, otherwise known as how CADM2 regulates preference and consumption of THC and cannabis oil-infused cookie dough in mice. So cannabis use has a significant heritable component. It's estimated that about 50% in the variance of lifetime or ever use of cannabis can be attributed to genetics. When it comes to candidate genes that have been implicated in lifetime use of cannabis, GWAS or genome-wide association studies have identified over 35 candidate genes. Among these candidate genes, CADM2 or cell adhesion molecule 2 represents one of the most promising targets because it's one of the only genes that has been replicated across these studies, and 3 polymorphisms within this gene have been linked to the risk of lifetime cannabis use. So CADM2, as the name might suggest, is important for cell-cell interaction, specifically at the synapse, and it's also associated with drug use phenotypes, cognition and processing speed, metabolism, and neurotransmission. So speaking specifically to its function in neurotransmission, it's predicted that CADM2 is important for excitatory and inhibitory neurotransmission, which is significant considering that delta-9 tetrahydrocannabinol, or THC, the primary psychoactive constituent in cannabis, is known to alter excitatory and inhibitory balance within the brain's reward circuit. On top of this, CADM2 is highly expressed within the frontal cortex of the brain, this region being responsible for higher order thinking and function, which may explain in part why CADM2 is associated with phenotypes such as impulsivity and risk-taking, alongside its association with drug use, including cannabis, nicotine, and alcohol use. So it's our belief that if CADM2 polymorphisms are linked with an increased risk of cannabis use in humans, then it could be the case that CADM2 variation affects cannabis use phenotypes by impacting pharmacological, motivational, or rewarding effects of cannabis. And again, if this is the case, then a deletion of CADM2 in mice might alter preference for THC-containing edibles, as well as preference for other drugs of abuse. So to determine whether or not this is the case, we developed a novel edible preference test to use in male and female wild type and CADM2 knockout mice. So in this test, the mice are delivered 2 balls of cookie dough. One contains THC, whereas the other contains vehicle. And the mice are given this cookie dough first as just control, but then are given one of these cookie dough balls containing THC in escalating doses, with these doses given for 3 to 4 consecutive days each. The mice are allowed to consume this cannabis or vehicle dough for a period of 30 minutes daily, and then preference can be determined as the amount of the drug dough that's consumed over the total amount of dough that's consumed as a percentage. So when it comes to preference for these THC-containing edibles, we do see some dose-dependent effects wherein, if the dough or the dose is increased, the preference for the dough decreases. However, we also see some genotype-dependent effects, wherein these knockout mice actually show lower preference for the THC-containing dough than the wild-type mice do. This is very interesting, though, when we look at the amount of THC that's actually being consumed by these mice that doesn't have any genotype-dependent effects occurring. So where this preference difference seems to be arising from is the amount of dough that's actually being consumed by these animals. If we look at the amount of vehicle dough that's being consumed in this graph that's on the left, as well as the amount of control dough, so dough given without THC, either of the balls having THC, we see that these knockout mice are actually consuming more dough overall than the wild type mice are. However, there isn't a compensatory increase in the amount of drug dough that's being consumed by these animals, thus explaining why the preference for the drug dough is lower in the knockout animals. One thing that's really important to consider here is that cannabis and THC are not the same thing. Cannabis contains over 540 other phytochemicals, and they're known to have interactive effects with THC. So it was very important for us to look at, whether we can extend these effects to cannabis oil that we're seeing with the THC dough. So these results are currently in the works. So I am planning to run more animals in the future. However, so far we have seen when we give a THC dominant cannabis oil in one of these cookie dough balls, we are still seeing this genotype dependent effect wherein the knockout mice seem to have lower preference for this dough than the wild type mice. However, I'm showing you these results more because they're very interesting when we contrast them to our initial pilot results. This pilot was conducted on exclusively male and older animals than what we're currently running. And if you're looking at, for instance, the wild type mice, preference in what we're currently seeing is much higher than what we saw in the initial pilot. So this could suggest that there are some age effects at play here, but it could also be a potential for a sex effect, considering we're now using mixed sex animals. And this would be in line with what is shown in human observations, wherein most of the time males have a higher rate of consumption of cannabis products than females, except in the case of oral administration, in which females will consume more than males. So we broke down these results according to sex, again, keeping in mind that, we are still in the process of adding more animals into our cannabis oil preference test. However, we have started to see maybe the suggestion of a sex effect in our wild type group when looking at the cannabis oil preference. However, these effects, according to sex, don't seem to extend to this THC preference test that we're running. Again, maybe suggesting that there is some difference when it comes to preference for THC versus cannabis, and it could be stratified by sex as well. So taking those cannabis and THC experiments aside, one of the things we're also interested in is whether or not there's differences in these CADM2 mice for other drugs of abuse. As I mentioned at the beginning of my talk, CADM2 is associated with other drug use phenotypes, including nicotine use. And so to determine whether or not this is the case, we employed a condition-placed preference test that we've currently piloted and looked at whether or not there are any genotype dependent differences across these animals. So in this test, mice are delivered either nicotine or a vehicle vapor, and then immediately following that exposure, they're paired with one of two unique environmental contexts. And so if they receive nicotine on the first day and are paired with one environment, on the second day, they're given the vehicle vapor and paired with the other environment. And this pairing occurs over the course of several days. The expectation here being that if a drug is rewarding, as we know nicotine to be, that when the animal has access to both of these environments, it will prefer to spend time or spend more time in that nicotine paired environment than it did at baseline. Again, these are pilot results, but we have seen this occurring in the wild type and heterozygous mice, wherein they're spending more time in this nicotine paired chamber after exposure than they did at baseline. But we don't see this effect persisting in these knockout animals. And in fact, they even spend less time in this nicotine paired chamber. So this could suggest multiple things. It could be that, maybe the knockout mice have a locomotor deficit, so they're not crossing over between the chamber sides. It could be that the nicotine itself is aversive to these animals, or it could be there's an associative learning deficit in these animals, so they don't make the association between the nicotine exposure with that environment. Just to address the first concern, it doesn't seem to be the case that there are locomotor deficits in these animals, as the knockout mice have no differences in locomotion compared to the other genotypes, and in fact may even have a hyperlocomotive phenotype. So one of the other concerns was that there could be an associative learning impairment in these animals. And to determine whether or not this is the case, we've again piloted this fear conditioning test in these animals, a contextual and cued fear conditioning protocol. So in this test, it occurs over 3 days. In the first day, the animal is exposed to an environment, and they are exposed to a cue, in this case, a tone, and then that tone is immediately preceded by a shock. And this is, pairing occurs over several times in a trial with the expectation that the animal doesn't like this shock. They're going to show freezing behavior, anticipating a shock arriving, and that's exactly what we see to be the case with the wild type and heterozygous animals, though the heterozygous animals to a lesser extent, but we don't see this in the knockout animals as strongly. Similarly, on the next day, 24 hours later, we re-expose these animals to that same environmental context in which they were shocked, with the expectation that the animal will have remembered they were shocked here and they will show freezing behavior. We do see this again in the wild type and heterozygous animals, but not in the knockout animals. And then finally, on the third day, we look at the response of these animals to just the cue. So these animals are placed into a totally novel environment where they haven't been shocked before, so they shouldn't be expecting to be shocked. However, we reintroduce that cue to them that was presented on the first day, expecting that the animal will have remembered it and will show freezing behavior in response. And again, we see this with the wild type and heterozygous, but not the knockout animals. So in combination with that, those results from the condition-place preference test, we are seeing potentially an associative learning deficit in these animals, though again, this is just a pilot, so we will need to run more animals to see whether or not this is actually the case. And on top of those potential associative learning and condition-place preference deficits, we also see these preference differences for THC and cannabis oil containing edibles that may have a sex effect also underlying that. But with that, I would like to again thank the IBANGS coordinators for putting all of this together. I'm very happy to be here talking to you all today. Thank you to my advisor, Dr. Jabran Kokar, who unfortunately couldn't be here today, but I know he's here in spirit. And of course, thank you to our collaborators at the University of California, San Diego, Dr. Abraham Palmer and Dr. Sandra Sanchez-Royce. And if there are any questions, I would be happy to take them now, or you can email me at thorpeh@uofguelph.ca.
Thank you, Haley. What a great talk. We do have some questions. The first was from another speaker in our session, Alexandra Gauthier. This may be a naive question, but what led you to choose cookie dough as the vehicle for THC delivery? Is sugar content a confounder?
So the reason we chose the cookie dough edible is because compared to other self-administration methods, typically in the past, they have been injection, so self-administration via mini pumps through a lever press, but those really aren't representative of how people are actually delivering THC to themselves. And there are other methods that we're currently, investigating or interested in doing, such as a vapor self-administration protocol. But we chose edible for a couple reasons. One is the practical reason of, we're still waiting on those vapor chambers. We'll hopefully have them soon and be able to run that test. But also there's a couple papers published by Smoker et al. In the last year where they did this cookie dough administration protocol. And they showed very robust consumption of products containing THC when it's delivered in this cookie dough form. Oh, and sorry, you mentioned also the sugar content. It definitely could be a case that sugar is a confounder here, especially considering, the link of CADM2 with metabolism. So that's why we're also interested in looking at other self-administration, such as the vapor chambers.
Thank you so much. So there are additional questions that we don't have time for right now, but I would refer you to the chat if you'd like to answer them, or if the people asking questions want to send an email. I believe Haley's email has been posted in the chat now. I would like to apologize to everyone. Haley, you did an excellent job considering you weren't set to give the first talk. I was looking at a draft version of our schedule. So I'd like to apologize to our next speaker, Wan-Her Lee, from the Rockefeller University. And she's going to be talking about the effects of